Okay, I think we're good with having the recording running. So like I mentioned to the few others who already were present, what I'm planning to do today is, of course, go through the material that relates to this week's chapter on the blood vessels. But I also want to take a little bit of time. Hi, Savannah. I want to take a little bit of time on looking at few things about the exam and practical that we had, and address any other questions that you might have on mind. But if at any stage you have questions, you want to ask anything, don't feel you have to wait to the end. Just feel free to shout out or write to the chat. I normally am able to see the chat, but once I start going back and forth with different screens, it usually disappears somewhere there. So don't feel you have to wait till the very end. I'm expecting that it's going to take a little over an hour because it's going to take just this little time for us to go through all the material in terms of the exam that I wanted to share with you. But let's start by having a look of the what I really had lined up. Our main focus will be on the covering the lecture material, but the two things that I do want to talk, I want to talk about the exam one and lab practical one. And I also want to address the last week's labs, because I know some students were able to complete those, some weren't, and I don't want you to feel that you're left hanging. The reason why I've been holding back from sharing what to do if you weren't able to complete those is that I wanted to make sure that everyone gets treated the same. So, kind of jumping back, looking at the exam and lab practical, most students should have their grades up there, and there's few that I'm still finalizing. I go through all the grades about all the answers, every single question about 3 times, and without knowing who the student is, and then they get finalized. The idea is that it's kind of graded in a constant same level of rigor. So if your grades are not yet up, you're one of those very last ones that have yet to do the 3rd cycle. But all of the lab practical scores or lab practical grades should be up. Exam grades, if your one isn't up, I'm expecting tonight, within an hour or so from our session, to have them up. And I'll make an announcement of that as well. But that's the reason why I wanted to talk a little bit about the exam. Overall, really, really strong scores. What students normally see that their grade moves up approximately 1 letter grade during the manual grading. If you feel that it really didn't work your way, you didn't get the score that you were after, I am open to opening up them so you can tackle those again. But that's something that we need to discuss a little bit together before I just open it up for you. A couple of general hints that I want to share with everyone kind of as a class. I think overall you were all onto it, but so no one feels that they were left out. If I share that there's going to be extra credit question in form of a short answer question, kind of a mini essay, that's up to you if you want to do it, or if you don't, I'm not gonna be offended if you don't do, but it's always a good chance to earn extra points. Usually I allocate about 1 letter grade's worth of points for those. I also encourage you to try to answer something if you can, especially on multiple choice ones. What's the worst you can do? You just don't get points for that. I don't do negative grading, so answering is better than not answering anything. So by chance, you might get it right. And I encourage you also to remember that it's open books, non-proctored exams for all of the tests, exams and lab practicals in my class. So use that, use your resources, use your notes, and make good notes. So let's have a look of a couple of things from the exam one. I picked up kind of example questions of things that I wanted to highlight. So multiple choice questions can be a little bit of a tricky field at times, because there can be a part of a multiple choice question that you immediately spot that, oh yes, I know what's the right answer to that. Make sure that you read through all of the answer options so you get all the parts of the question correctly. For example, here where I was comparing the exocrine and endocrine, I asked about whether there's a duct or not, and then I asked about whether it acts locally or through all parts of the body. So it was easy to get kind of sidetracked if you just went with the first answer that you saw. So make sure that all of those gaps are filled in a way that makes sense. Also, true and false questions. Back in the day, I think students thought that they're the easiest questions to tackle because you have 50-50 chance to get it right. I actually think that they're often the hardest questions because a lot of the time true and false questions are formulated in a way that the first part makes sense, and there might be just the tiniest part that makes it untrue by having been added there. For example, in this example, I was asking for two hormones that are oxytocin and prolactin, and telling that they are both secreted from posterior pituitary. Well, that was the trick there. I think that the prolactin is secreted actually from anterior pituitary. So all other parts of this sentence made sense until you got to the very end, and that's where the trick was hidden. So just be aware of that. Sometimes true and false questions can be a little bit mean like that. It's also really easy to get mixed up on the true and false questions in terms of that just a single word might be the difference. So here I talked about medulla of adrenal gland, whereas it was actually that the medulla secreted the epinephrine. That part of it was true, but the cortex was the part that secreted the 25 other hormones. So that was just hidden in there. So just some things to keep on eye. In terms of there's sometimes specific combinations of things that you can kind of learn when you're revising the material or making your notes. So for example, on this question, it was straightforward. What is the hormone that the kidney acts through to regulate blood pressure? And in terms of the kidney, it's going to be the renin, but there were other options, I think angiotensin and so on, which play part of that pathway, but the renin is the main. So just kind of try to make up those connections of whatever makes sense to you, that kidney always impacted by renin impacts blood pressure. So that's one way to tackle those, what to remember and how to remember them. I do think that one that often catches on multiple choice questions is if there's options that sound very similar, because we have a tendency of looking for questions, quickly moving on, and if we pick an answer and don't really read it carefully, we might go for one that sounds correct but isn't exactly. So in this example, for example, we were talking about melatonin, which was this hormone that had played role in controlling the sleep-wake cycle. Well, there was also melanin as answer option. Well, melanin, you remember from the chapters when you covered in BIO201, the pigments of the skin. So it's something that's familiar, but it's not exactly what we're looking here. So just something to keep on mind. Sometimes you can also wonder, is it too simple question, or can it be that simple? And usually people get super confused that, oh, there should be a trick somewhere. So in this one, the simple statement was that thymus would be a very pronounced gland, but the key there was that in adults, well, we remember that thymus shrank over age. So that was a false statement, but just those two words there kind of made it false statement. sometimes there can be very complex questions sneaked into a multiple choice format. So, for example, when we talked about water soluble and lipid soluble hormones, of their physiological actions, that the water solubles act on the cell membrane and then need second messenger to act within the cell. Whereas the lipid-soluble ones were able to pass the membrane, go inside the cell, and activate the gene directly. So even though it's a single question, it can contain a lot of information. So bear in mind that sometimes some of those multiple choice questions are structured a little bit like that. If there's something that I highly emphasize in a class, it's probably going to come up. And one of those has been the concept of homeostasis. So if you hear it more than 3 times in a class, probably worth of making a note. It's going to come up one way or another. And there were a few different questions about the homeostasis. It was the definition, but it was also this diagram that I introduced to you. I know that not everyone has taken the intro bio classes with me or the BIO201 with me. So it might feel like, oh, why are we going to something that's that simple even this late? Well, it's because, like you'll see, for example, on this week's chapter, we use this very same diagram to talk about other processes. So if it's something that you keep hearing time and time again, it's worth of tackling that. There's also questions where you might end up finding only few examples of opposite reaction. So, for example, often we've noticed that negative feedback was what we most often saw on the homeostatic feedback mechanism loop. But the only exceptions of that where we saw positive feedback were birth, lactation, and blood clotting. So if you knew the positive ones, just click away all others that, oh, they have to be negative examples. So just to show you how these questions are often formatted. If it is something that, again, I mention a lot of the time, the terminology plays a role on this field. So we talk a lot about this poiesis being a production process of something. But then I mentioned that there's different things that we can produce. We can produce blood in general, which was hematopoiesis. Erythropoiesis was the production of red blood cells, whereas leukopoiesis was the production of white blood cells. So if you keep hearing that a term comes up, do make a note of that. That might be something that comes up. Also, numbers are usually quite important on these fields where you're heading. So kind of having an understanding what is common, what is not common. So, for example, here we were looking at the blood composition where about half, a little over half, 55% of the blood composition is plasma. Then 45% red blood cells, and white blood cells and platelets are less than 1% each. So again, when you see that certain things come up, they make very good exam questions. So that's just kind of to get your mind thinking what you might face. There are some themes in anatomy and physiology that are fundamental topics. So I would assume that your BIO201 courses started by talking that the form and function are related, that the anatomy defines how physiology happens, or physiology might define an anatomy of the structure. And these very core concepts we can apply to almost any chapter that we look at. So in this question, I was just applying that very core concept to talking about the shape of the red blood cell and how then that had an effect on its physiological properties. So just something to bear in mind. I also shared with you that in the field of anatomy and physiology, we love mnemonics. So there was a question that I think would have been a harsh question otherwise, listing the different white blood cell types from most common to least common. if you had not been given a mnemonic for it, which was never let monkeys eat bananas, if you remembered that you didn't even need to remember the names of different white blood cells, you just organized them in based on that mnemonic. So keep an eye when things like that come up. In terms of the lab practical, there again, I think that was more options that were acceptable than what the automated grading said. But for example, here, it's really good idea to be very, very, very precise what it is that is being asked. So of course, we see here pancreatic islet cells, but we were looking for 2 cell types. So in that case, it's going to be the alpha and beta cells. So if you told me that, oh, it's pancreatic islet cells, you could get partial points, but for the full marks, you needed to talk about those 2 types of cells. So always read the question really carefully so you get those correct. Also, what is precisely the structure that you're being shown to? So there we had parathyroid glands, which are located within the thyroid gland. But you kind of are not going to know those unless you read the question very precisely, look very precisely where the arrow is pointing. Same was with this one where we saw the adrenaline gland, and the question was about the black arrow where it is pointing. Of course, it was pointing to the medulla, but when I first looked at that myself without actually reading the question, of course, my eyes could be drawn into this box. So I'm not surprised that a lot of people told me about cortex of the adrenaline gland, but just read the question very precisely. Also, if you're totally lost, look, can you see anything else in that picture? Some students got confused and couldn't tell what gland it was, but you noticed right here at the corner, there was actually a diagram that showed the kidney and the adrenaline gland on top of that. So I would never ask you something that you wouldn't have a way of knowing what gland it is or what structure it is. I try to give some sort of clues. Also, if you can provide me additional information or more precise information, you might end up with extra credit. So if you see on your notes or on your feedback EC written, that's often extra credit. So here I was asking about the portion of the gland indicated by the black arrow. So the portion was, of course, head. But if you told me that it is head of pancreas, you actually got extra points. So that's always worth of grabbing those as well. Here I have a similar example, giving the full information or precise details. So we had a picture that was from pituitary gland, but it was especially from the posterior lobe of the pituitary gland. So if you gave me both of those, you ended up with extra credit because you gave a little more than the minimum that was being asked for. Same was here then for the anterior pituitary gland. So just kind of to show how these questions work and how the grading works. You'll notice I'm very, very generous with the grading. I normally try to give points. If there's any way of giving you points, I'll give you some. Here, for example, you were being asked to identify the structure. If you don't know the structure in relation to the material that we have covered, for example, on this chapter, we had talked about different endocrine glands. So the hypothalamus was the structure that we saw here. But the truth is that that could have been just as well third ventricle. Well, we hadn't been talking about ventricles of the brain on this chapter, but if you knew it from the earlier on, from BIO201, I'm all game for that. You can give me answers from that, and that's just as correct. If you gave me both, then you would have ended up with extra credit as well. I also am aware that there's often structures where there's multiple names that you could use. For example, here, the red blood cells, RBCs, or erythrocytes. So any of those would have earned points. But then if you use the erythrocyte, which is the proper scientific term, you actually got a little bit of extra credit by doing that as well. So that's really how the exams worked. I encourage you to have a look through your exams, and I encourage you to take the time to read the feedback that I've written. Normally, I write more feedback if you get something wrong than I would write if you get it right, because there's less for me to say if you've got it, if you know the concept. I know that there's few of you who might not have yet received your exam one feedback. So if that's not up there, just keep an eye. Probably in the next hour or so after this session, it's going to show up there. But I want to tell you that I am not a dictator in terms of how I grade. If you feel that something doesn't quite make sense or quite doesn't add up, send me a message, because you'd be surprised how much mistakes your instructors do. And I'm not trying to badmouth anyone else. I'm not trying to badmouth that I wouldn't be paying attention to your work. I do, but we grade hundreds and hundreds of exams in one semester, for each exam being multiple questions. So there are oversights. So if you feel that something doesn't add up, ask, because more often than not, I'm like, okay, hey, I would take that answer as well when a student approaches me and asks. Also, if you feel that this really didn't work your way, I am totally open for considering should you resit that test. But just make sure that we have that dialogue together first before you ask for that to be open. So I take time this week and last week. I have actually met with quite a few students one-on-one. So just send me a message and I will make myself available for you. I usually am able to do that with a very short notice time. So that was about the exam and lab practical one. Like I said, really, really good standard of work. Many of you might notice that you can even get more than 100% in my exam or in my lab practical. That means that you have to work that much less in the future. So it is possible that by the time we reach the end of the semester, you can choose not to do something because you already have such a good score or such a good marks. So collect those points as much as you can. I guess that's what I'm trying to say. If you have any questions at this point about the exam or lab practical one, I'm totally open to answer those. If you want to contact me later, I'm also totally open for that. And having said that, if I don't hear anything, I'm going to move on. But don't feel that that's me avoiding your question. I'm just wanting to be respectful of your time. So last week we did the labs that were built on the old Flash format. So I know that Flash has retired and it works for some it doesn't work. If you have really new laptop or PC, that might not have worked. So I saw that most students were regardless able to complete those. But if you were not, I told you at the time that I have an alternative assignment. I just haven't posted it yet because I kind of wanted to see how it maps out and who needs that. But keep an eye for an announcement probably tonight or tomorrow morning where I will tell what you can do if you were not able to complete those. But as always, you can contact me directly with specific details of what the issue was. And I'll make sure to find a way that no one loses out on points. I'm not in a business of trying to make any of you not succeed. So that goes to also any missing assignments or retaking assignments. I always encourage you to try to take it again. So often I set my assignments in a way that you can submit late work and it actually doesn't deduct any points, even if it's after the deadline. Sometimes you're not able to do that and you might need to message me so I open it for you. But when you message, be specific of what assignment it is so I can kind of go back there and get you set up. But like I said, my goal is to get you to do as well as you possibly can. So don't think that I'm here to catch you out. I'll work with you to make sure you can collect all those points. The best strategy is really to stay on top of the work. So what we're doing this week, we're doing the same stuff that you've seen before, the LearnSmart, the quiz, and the practice anatomy. And actually the lab is going to go back to the format that you already knew on the publisher's website, on the Connect. So there's no field labs. It should run as it ran in the past. It should be quite straightforward. In terms of this week's material, what we're going to do, I'm going to go through it next. I do think that we're going to run maybe a little longer than what I have scheduled for this session. So if that happens, I won't be offended if you have to go, but I will try to go through this rather quickly. But I also will try to make sure that everything gets covered. There's always going to be the recording, though, available for you. So bear with me as I jump to a different PowerPoint. If you have any questions of what we've covered so far, or you have any questions that come up while we cover the material, just let me know. I really do want to make sure that you know that I will take the time to answer. So on that note, I think I have the presentation here, and just let me share it with you. We are gonna get it. It should be jumping up on your screen about now. So I'm just gonna give a kind of a quick walkthrough of the important points that you might wanna know about the blood vessels in relation to the new material that we're covering this week. So let's have a look at that together. And so far, it looks better. So far, we have talked about the blood, we have talked about the heart. So this week really wraps up the discussion on the cardiovascular system. Ooh, I didn't mean to go that way. So it wraps up the discussion on the cardiovascular system, but you will not be retested on the blood. You've already taken the test on the blood. And actually, your final will not go back to those things. It's not cumulative. It's something else. It's a project-based final. So if you've had it with blood, you never want to think of it again. On this course, you don't have to think of it ever again. So we will be focusing on the blood vessels. So kind of the tubing of this system on this week's material. It's kind of those channels or conduits where the blood travels, and the heart was the pump moving that blood within there. Of course, the relevance for us in a clinical sense, since many of you are thinking of a clinical carriers, it's all to do with bleeding, bruising, blood pressure being too high or too low, various blockages that can be forming within the blood vessels, and so on and so on. So there's many uses for this information in terms of when you move forward in your career. And when I ask students to list me what kind of blood vessels we have, this is usually what I get from them. I get them telling me that we have arteries, we have capillaries, and we have veins. And that's really good, and that's been perfect for this far in your studies. But since we are doing 200 level course, we're gonna do a little bit more. We can do more than that. We're going to add arterioles and we're going to add venules to this list. So you remember the arteries were the big blood vessels that left from the heart. Blood that's leaving the heart travels in arteries, where arterioles are just simply going to be a smaller version of those right before we move to the capillaries, which were the smallest vessels, and those were where the exchange of materials took place. And then once we have finished with the capillaries, we go to the venules and then we go to the veins. and veins were the ones that actually brought the blood back to the heart. So there's going to be 3 layers that all of these blood vessels, whether it is artery, arteriole, capillary, venule, or vein, they all share these 3 layers. Some of them have more prominent certain layers, some of them have less prominent certain layers. But we're going to look at the different types of blood vessels once we have established kind of a common general field of what layers we see on those. And these layers are known as tunicas. So I don't know how you would think of that. I guess that ladies often put tunicas on. It's some sort of a layer or outer layer of clothing. So if that helps you to think of that in that way. So there's 3 that we're going to look at. Tunica intima, tunica media, and tunica externa. And I think in the UK, you might often end up seeing that rather we talk about tunica adventitia, but it is the same thing. So if you see those 2 being used interchangeably for the last one, don't let that throw you off. But we'll go through these 3 layers, 1 layer at a time, looking at what are the characteristics of each. And we're going to go through them in an order that we're going to start from the middle of a blood vessel and work our way out from there. So the middle part of the blood vessel is known as the lumen. So we're working in that order. So let's start with the tunica intima. So tunica intima is the innermost layer. I think of it that it's in an intimate contact with the blood inside that blood vessel. So if this is the inside of the blood vessel and this is the blood, it's in the most direct contact with the blood vessel or blood inside it. Whereas the other layers, as we continue, they're not in as intimate contact. So what else could I say about the tunica intima? We notice also that it's made of these cells that we see here. And they look familiar from what we have learned in the past about the endothelial cells. So these are simple squamous cells. So simple meaning that there's 1 layer of them, and squamous that they're kind of these flattened discs in a cross section. Well, this actually makes sense if we think of why are they that shape, considering the function of the tunica intima. The function of the tunica intima is really to protect these other layers that we'll talk about, but also facilitate easy passage of materials from the blood to the outside of the blood vessel. So we want to keep that layer thin. So that's why we have this only simple layer and squamous cells, which are easy to cross. Well, that's our tunica intima. Let's have a look at the tunica media. And tunica media is going to be the middle layer. So that's the thickest layer of all of these. And it's largely made by smooth muscles. So you can see on this microscopy image the smooth muscle. Layers there. And that tells us the smooth muscle of what actually is going to be the function of the tunica media. The big function of the tunica media is going to be to control the diameter of these blood vessels, where tunica media would be prominent. So we heard about the term constriction and dilation already earlier on in previous chapter, but just as a reminder, when we have vasoconstriction happening, the diameter of the blood vessel goes down. When it's by vasodilation, the diameter of the blood vessel increases. So tunica media with its smooth muscle layer is responsible for that. And like I said, it is the thickest layer that we end up seeing. There's going to be some amount of elastin as well, but really if we think at the function, well, because of the smooth muscle, it is all this contraction and dilation that the tunica media focuses on. Then the last one that we have to look at is gonna be the tunica externa. So sometimes known also as tunica adventitia. So what tunica externa does, it really is, sorry that it jumps ahead. Tunica externa is made up largely of connective tissue. And its function is really then to support the blood vessel by reinforcing the walls, by anchoring the blood vessel to its site. There's also going to be some nerves, some lymphatic vessels running in there. But if we now think of that, what we learned about the heart in our previous chapter, we learned that the heart itself and the heart muscle, just like any other tissue in the body, required oxygen and required nutrients. Well, the same is going to be true for blood vessels, and especially if they're big blood vessels. Tunica externa, again, we remember it's the outermost layer here of the blood vessel. So it's far away from the blood inside the blood vessel. So for that reason, it might be hard for this layer to actually get blood from the inside of the blood vessel. So we might need to do these special adaptations, which are little vessels coming out of the blood vessel, supplying the wall of the blood vessel itself. So tunica externa sometimes receives blood supply with these tiny vessels that come out from there. And these are known as vasa vasorum. So they supply the blood vessel wall itself, nourish that. So that tissue remains alive. Well, really, this next image is not something that I want you to worry too much about. I just simply want to show that we see all these three layers. Tunica intima, this innermost layer that we saw earlier on, which is simple squamous endothelial layer. So single layer of these flattened disc-looking cells forming the innermost layer, which played the function of protecting other layers and facilitating the passage of the materials. We also see the tunica media, and tunica media was, of course, this smooth muscle layer. So this thick layer of smooth muscle, and its function was the vasodilation and vasoconstriction. So controlling this diameter of the blood vessel. And the final layer that we saw, tunica externa, that was all about anchoring the blood vessel on its side, supporting it in terms of giving its further structure. So we see these three layers, both in arteries and veins, and also in arterioles, venules, and capillaries. So those are the three layers that I wanted us to be familiar. So now that we shift our focus of looking on these different kinds of blood vessels that we have, that you're able to see that these differ in the relationships of those. So let's start with the arteries. And what you need to know about arteries is that, of course, arteries were the first blood vessels that we had after the vessels leave the heart, after the blood leaves the heart. So they are under a significant pressure and a significant blood pressure. So the heart pumps the blood out from the left ventricle to the systemic circulation with a significant force. And it is these arteries that have to deal with it. Also, remember that not only it's a high blood pressure, it's also very pulsatile in its nature. So heart doesn't pump blood out on a constant rate. It was when the left ventricle contracted, there's a huge shooting of the blood, and then there's a relaxation time of the left ventricle when the blood pressure drops. So these arteries have to be ideally adapted for that. So that's two things that we need to keep in mind regarding. So their walls are gonna be very elastic and have thick walls. So really the function of arteries, if you want to summarize them somehow, is to kind of smoothen this flow of high blood pressure in a pulsatile manner. What we see on this little picture here, it's a microscopic image of an artery and a vein. And if we look at this artery, we see this blue layer. That's the connective tissue layer there. And that's what gives the artery its strong shape. So even when a person dies and we take away all the blood from the body as part of the embalming process, the arteries remain nice and round in their shape. Whereas the veins, as you can see on this microscopic image even, they start to collapse. Their walls just don't stay open, nice and round. So when I work with dead people, dead catheters, and I have to tell apart an artery and a vein, and I don't want to follow the origin or the destination of that blood vessel, I just simply feel it. And arteries feel much more rigid, whereas the veins kind of squash when I squeeze them. So that hopefully helps to kind of reason why do we see and what do we see with the arteries that makes them special. So let's have a look of the three different kinds of arteries that we need to talk about. And the first one is going to be the elastic arteries. So here's a nice picture of an elastic artery. So you'll notice that they have a very thick wall here. In addition, this lumen, the middle part of the elastic artery, is really focused on facilitating the kind of flow of material. So there's a low resistance for the flow of the blood along this vessel. It's not really that worried about moving the blood across the artery, the elastic artery. We're just wanting to conduct the blood to the areas where they're needed. Really, the passing of the materials is not a great concern for us. The thick characteristic of these elastic arteries also tells us that these act as these pressure reservoirs. And by that, I mean that they do kind of try to stabilize this blood pressure variation, this pulsatile flow, so that when we pass that blood forward, that variation kind of decreases as we move further in the vascular tree. A good example of an elastic artery would be, for example, our aorta. So there's a lot of elastin, as the name would suggest. And there's really no vasoconstriction at all. The blood vessel itself doesn't work to constrict. It simply expands and recoils back to its normal shape as the pressure fluctuates due to this pulsatile flow. The next one that we're going to have a look of is going to be the muscular arteries. So here we have a very nice picture of a muscular artery. And they are often described as being distributing arteries in comparison to this conducting arteries. So conducting arteries simply transport the blood from one place to another, whereas distributing arteries start to then spread it to various directions where they are needed, distribute it to the points where they might be used. So most arteries that we end up finding in the human body and most arteries that we name and we study are going to be these muscular arteries, these distributing arteries. They have the thickest tunica media layer with the lots of smooth muscle, a little elastic tissue. So these are able to do vasoconstriction. And now if you think of why this could do vasoconstriction, but the elastic artery couldn't, think of the function of distribution. When we are choosing where the blood needs to go, We want to make certain blood vessels larger and certain smaller so we can choose where that blood, majority of the blood should go. Whereas with elastic arteries, we really are not concerned. It's just one tube to another place. It's not doing any distribution. It's simply conducting it from one area to another. I hope that kind of makes sense in explaining the difference between the two. The final one that we have here is going to be the arterioles. And really, we could look at arterioles as a group of their own. So eventually, they are just these small single layers. So we don't end up seeing any other layers there on arterioles than just the tunica intima. So only this simple squamous endothelial layer is left on the arterioles and at the very end of the arterioles. This is because this is right before we reach the capillaries. But you might be thinking, okay, why do I care about this? Well, arterioles play a especially important role in choosing to what capillaries blood goes to and what capillaries blood does not go. So what we see here is a bunch of capillaries. And you'll notice when you study the physiology, and especially the numbers of the physiology in terms of the vasculature, that we actually have a huge amount of capillaries in the body. And we have so much capillaries in the body that if all of the capillaries were open for the blood to flow in, we would run out of the blood. So we always have to choose that blood goes to certain capillaries and certain capillaries are blocked from receiving blood. And this is done by these little structures known as precapillary sphincters. So let's have a look at those. See, these are muscle bands within the arterioles right before an arteriole changes into a capillary. And these muscle bands are able to open and close these different capillaries. And whenever we have this kind of a network of capillaries, we talk about a capillary bed. And in this diagram, we see this whole capillary bed here being open because all of these precapillary sphincters let the blood to flow into various of these capillaries. Whereas here, these sphincters are closed. So here we simply want the blood to go through this area but not spend time in. So this is how we choose that what areas of the body receive the blood and what areas do not. So for example, if you're ready to do some physical exercise, your skeletal muscles start working hard, well then you do want the capillary networks that supply the skeletal muscles to be open by these capillary sphincters. But if you're exercising hard, you're probably not going to worry that much about producing urine at that time. Your focus is elsewhere. So capillary networks that would supply urinary system are probably going to be closed. So that's the function of these precapillary sphincters and why they are such an important part of the arterioles, choosing which capillary beds are open. So that completes what I was going to say about the arteries and moves us to talk about the capillaries. So let's have a look of the capillaries. And what capillaries do, they were the main site of exchanging material. So these are the microscopic small vessels. They're actually often even a little bit smaller than a diameter of a single red blood cell. But red blood cells can still pass through those because red blood cells, because of this biconcave shape, can actually squeeze a little bit. And the only walls that we end up finding in capillaries are gonna be the tunica intima. So that's the only layer that we end up finding there. So these capillaries are the site where the exchange to the cells on that area or interstitial fluid takes place. So we, for example, give away from the blood oxygen and nutrients, and we pick up from these cells or interstitial fluid carbon dioxide and waste products. So it's all about this exchanging of materials and passage of small materials through those. So there's going to be 3 types of capillaries that we're going to look at. And I'm just simply going to really quickly jump out of here, and not because I'm covering this, but I noticed that my slides keep jumping ahead, which is not something that I want to do. So let me just fix that. I hate it when I don't have a control over my slides. This should fix it. Now we should be able to continue without them jumping back and forth. You notice that I keep fighting against it. So let's go back. So there were 3 types of capillaries that I wanted to speak with you. Well, they are going to be named continuous, fenestrated, and sinusoidal. And you might initially be like, oh my gosh, it's another 3 names that I have to remember. But those names actually explain exactly what these capillaries are like. So let's have a look at those. And we're going to start first with the continuous capillaries. Here I was thinking happily that we're done with them jumping around. I think that was just my fault. So continuous capillaries, if you look at the endothelial simple squamous layer that it's made of, all of these cells are connected to the next cell. So they form a continuous wall. That wall is continuous. There's no breaks. So that's where the name comes from. And we end up finding continuous capillaries, for example, at the skin, muscles, lungs, central nervous system, and also the separation between the blood-brain barrier. So we don't let all the blood that circulates in the rest of the body to pass to the brain without any control, because brain is so sensitive. We don't want any toxins there. We don't want any drugs there. So there is a filtering layer there, and that's made by these continuous capillaries. So continuous capillaries, cells are continuously connected to each other. The next one is going to be fenestrated capillaries. Well, term fenestration, I think it comes from French, which I might be wrong about, but it comes from some language, and it means a window. So their fenestrated capillaries have little windows in them, and through these windows is where the exchange of material happens. So these are especially good for filtration, absorption, and secretion of materials. We find them, for example, at the kidneys, intestines, places where we have hormones being secreted, and so on. So really, I kind of describe them, they look like Swiss cheese. So these walls have those little windows, fenestrations, little holes. So you can imagine what the last type of capillaries is going to be. That's going to be sinusoidal capillaries. And instead of small windows, there's actually huge parts of the wall missing every here and there. And in addition to allowing a lot of exchange between the capillary and outside of it, the sinusoidal capillaries also cause for the blood that flows within them to slow down. When you have large parts of the wall missing, that slows down the flow of the blood within those capillaries. So they're really, really good for clearing blood, for example, in liver, bone marrow, spleen, adrenal medulla. Those would be places where we would see sinusoidal capillaries. And that's all you need to know about the capillaries, really, that I can think of. And the last one that we're going to have a look of is going to be then veins. And before veins, we of course have our venules. So venules are just smaller versions of the veins after the capillaries. And then as we continue further along, we reach the veins. Well, what's the function of the veins? Veins bring the blood back to the heart, and now this blood will have little amounts of oxygen, whereas the arteries had high amounts of oxygen. The other thing that we notice, which is quite interesting about the veins and quite important for what we're going to be talking about next, is that the blood pressure within the veins is very low. So there's a low blood pressure in the veins. Their walls were thinner, like we saw at the very beginning, and actually most of our blood vessels in the body are veins. So lots of the blood is actually at one time within the veins. So then I ask my students always that, okay, let's think of heart being at this level of the chest and blood flowing from the heart through the arteries, through the aorta, and then arterioles, all the way to the capillaries of your toes. It makes sense that this blood is pumped with high blood pressure and it flows nicely there. But how do we get that blood back to the heart after capillaries from your toes back to this height? That's going to be a challenge. I often hear many different kinds of answers from students. Some students tell me that, oh, it's because of this force, how the blood was pumping blood out so that new blood is pushing the blood to the veins and back to the heart. That's not the case. I also hear that, heart sucks the blood back in. That's not really the case either. The return of the blood to the heart through the veins is actually a really clever adaptation. And that shows us why human body is so cool. So what we end up noticing and what we already know, that the veins had thin walls. And there's a reason why their walls are very thin. Well, just as you remember when we talked about the heart and how we had different kinds of valves, or we had many valves there separating the various chambers, we actually end up finding those valves also within the veins. And the function of the valve was, of course, the unidirectional flow of the blood, only to one direction and not to the other direction. Well, we end up finding those valves in the veins as well. And what we also end up finding that a lot of veins get squashed all the time. And as they get squashed, this blood gets pushed up or down to some direction from that place where it gets squashed. But it never continues to the wrong direction because of these valves. So blood has only one direction to go to, which is towards the heart. And when that squeezing stops, yes, some of the blood would fall back in, but there's going to be valves that stop it from falling back in. So that's why we're actually constantly moving a little bit. It can be us moving physically, our skeletal muscles squeezing these veins. It can be also our breathing, for example, in thoracic cavity, squeezes some major veins in there. Even when you're sleeping, you're constantly moving a little bit. And all of this has to do with getting this squeezing of the veins that returns the blood to the heart. And this is known as the venous return. Well, good way to understand what happens here is to think of a situation where we would not be moving, which would mean that we would not be getting blood going back to the heart. That would not be great. If we don't get blood going back to the heart, we also don't get blood going to the brain. We're running out of blood to pump, and your brain will not tolerate being starved of oxygen because there's no blood coming back. So let's see what would happen. This is what happens. You end up passing out. So if you look at military parades where soldiers stand for a long period of time, totally still, well, now their veins are not getting squashed. This blood is not being pumped back to their heart. And we're running out of blood within the heart to pump it to the head and pump it to the brain. Your brain says that, oh, this is not good for me. I'm soon gonna get damaged. What's the easiest way to fix that? Well, get that soldier to lay down. And that's exactly what we do when someone passes out. Let's lay them flat horizontally when it's easier for the blood to travel and travel also to the head. So that's why soldiers end up passing out if there is no venous return of the blood to the heart. And that's why the circulation to the brain is also affected. There is another clinical relevance, including with this venous return. Think of if some of these valves would happen to fail. What would happen is that more blood would build up underneath it where the next valve would be, and the vein would end up swelling on that part. That's because the veins had soft walls. They were not very rigid. Well, this is how the varicose veins or spider veins end up forming. So what we're seeing is basically part of a vein above which the valves have failed. So blood has started to pool into this one location and accordingly then swell the vein wall. What causes varicose veins? There is some amount of inheriting it from the parents' tendency to that, but also there's certain conditions where your venous return is poor, high elevation, high pressure in the veins. So that's why veins really should have a low blood pressure in. So that's how varicose veins are formed, and I hope that helps to kind of explain how the venous return works. I realize that I'm going through all of this really quickly, so if you feel at loss, ask me, or also look through that full lecture video. I kind of spent more time on those covering these points. So the next thing that I want to move on, we kind of now had a look of the more anatomy and what are the consequences of the anatomy. I now want to move our focus on the physiological aspects. And the first aspect of physiology of the blood vessels that I want to look with you has to do with the flow of the blood. Well, flow is measured as a unit of something within a period of time. So in this case, milliliters in 1 minute. How much blood flows through a certain location? That's flow. That's a simple concept. The next concept that I actually really want to focus with you guys is a little more complex, but it's much, much, much more important as well. That's going to be our blood pressure. And if you think, how would you define what is blood pressure? Well, blood pressure is, if this is our blood vessel, it's the amount of force that's being excreted against the wall of the blood vessel from the inside of the blood vessel. So that's often measured as a unit of millimeters of mercury. That's a force, measure of a force. And when you ask from someone that, oh, what's your blood pressure? They will typically give you two numbers, an upper value and lower value. So you might wonder, why do you get two different values for a pressure? Well, let's go back and remember that we typically measure the blood pressure from the arteries. And arteries receive the blood from the heart in a pulsatile manner. So at times, it is when the left ventricle contracts and the blood flows out at the high pressure. And then there's going to be times when the ventricle is relaxed, blood is still going on the veins, but it's not at quite as high pressure. So this, when there's more pressure, when the contraction of ventricles takes place, that's known as our systolic blood pressure. And the diastolic blood pressure was during the period of when the left ventricle was relaxed. There's a lesser blood pressure. Typically, if you ask for a person's blood pressure, you get something like these numbers, 120/80 or so on. Those numbers vary a little bit, and what's a normal blood pressure varies a little bit from year to year, depending on the recommendation guidelines that we use. So, that was the explanation of the systolic and diastolic blood pressure. And when we try to work out what factors would then affect your blood pressure, there's multiple things that we'll notice. Of course, the total amount of blood in your cardiovascular system. So for example, if you start bleeding a lot, you will lose some blood. The total volume of the blood will decrease, and as a result, your blood pressure will drop. So that's why if you have someone who's been losing a lot of blood and they get to the hospital, we tend to try to get their blood pressure up by giving them more blood. That can be either in form of just simple plasma or a blood transfusion. The second factor that plays role to the blood pressure is going to be, of course, our cardiac output. And we talked about the cardiac output actually in our previous chapter. We finished with that as a measure of how much the heart contracts, how much blood the heart pumps out with each contraction, and how many contractions do we have in a minute. So that was the cardiac output, how much blood is being pumped out from the heart. The third factor that affects blood pressure is something known as peripheral resistance. This is the resistance to the blood flow within the vasculature. So it's not easy, it's not totally without resistance that the blood flows within your blood vessels. There's some amount of resistance for that, and the amount of resistance is known as peripheral resistance, or PR as an abbreviation. And of course, then the factors such as how well the blood flows in the vessels, how elastic the vessels are, and so on play also a role. So you can really calculate actually your blood pressure by multiplying the cardiac output by the peripheral resistance. So that's what we kind of have a general idea what the blood pressure is. Well, some individuals, when you go to a doctor and we measure your blood pressure, we end up finding that they have unusually high blood pressure. And they're like, no, my blood pressure normally isn't this high, but because I come and see you, my blood pressure jumps up. This is known as a white coat syndrome. It can be either because of a doctor or a nurse measuring your blood pressure, or being in a clinic or being in a hospital, which is making you nervous, and your emotions have a significant effect on your blood pressure. So that can bring up your blood pressure as well. And what we tend to do normally, we try to measure your blood pressure from the level of your heart. We try to have the patient sitting down for a while before we measure it. So next time you go to see a doctor or a nurse, notice they sit you down for a second before they actually get on to measuring your blood pressure. Clinically, we should really actually measure your blood pressure 3 times and take the middle value or take the average of those. Ideally, if you do have white coat syndrome, we give you or tell you to get a blood pressure cuff to home. And then you measure your blood pressure at home where you're relaxed and comfortable. And so that tells us more realistically what your blood pressure is more likely to be. Well, blood pressure and pulse are related. So what pulse is a kind of an indirect way for us to tell your heart rate. How many times your heart contracts per minute. This might be a useful number for us to have, but of course we don't want to open anyone's chest just to see that. Well, it turns out that we can actually feel it by feeling some of your arteries, that how many times we get this pulsatile action of the systole and diastole of the blood pressure in those. And because if you feel the arteries, you will feel very strongly this pulsatile flow of the blood still right after the heart. And that's a good way for us to get the information about one's heart rate. Typically, that's somewhere between 60 to 100 beats per minute, varies from individual to another, varies based on many factors. Also, athletes, I used to work a lot with athletes. Some of them have a very low resting heart rate. I had some that were like 30 or 20 beats per minute. That gets kind of creepy when you measure that. But that's just because they're so primed to the extreme in their body. So here's a couple of our multiple places where we can measure the pulse. Probably the most common one is to take the radial pulse, but there's many other locations, as you can see, where in the body we can take the pulse from. So it doesn't just have to be from your wrist. It's based on where there is a need. But we should try to measure it from the same side if we're kind of keeping a record of that. And what's important is that we know individuals' normal blood pressure and normal heart rate, normal pulse. So that's why the nurse, whenever you go to a doctor, records those. So we have some sort of baseline against which we can compare then your current condition. So that brings us to talk about how do we regulate the blood pressure. And we're going to start by looking at the regulation of the blood pressure in short term. And what you'll notice, we've got this nice diagram of homeostatic feedback mechanism loop that I keep talking about, and it's going to be really important for us. Well, this is the time when we see that it's a great way of explaining short-term regulation of the blood pressure. So let's start with the stimulus, which is a change in your blood pressure. Well, you remembered we had a control center, and within the control center, we had some sort of a set point, range of normal values. And normal blood pressure could be, for example, 120/90 or 120/80. And if it's anything off from that, if this stimulus, this blood pressure is anything else than this set point, then we are going to react. Well, let's imagine that it is going to be off from that. How we are going to notice that is through baroreceptors or barochemoreceptors or chemoreceptors. These are little receptors located in various parts of the body. For example, within the carotid sinuses, which are these dilated areas, for example, where the common carotid artery divides into two, there's a little bit of an expansion there, and that's where we have carotid sinuses. We also have these at the aortic arch, so the arch of aorta, where the big vessels come out. We have receptors there, and also in various different large arteries, especially at the neck and thorax. So those would tell us, those receptors, they would sense that the blood pressure is off from what we normally want it to be. This message gets passed to the brain. We remember brain was the most common control center in human body, and especially to the part of the medulla or medulla oblongata, if you want to use the full name, within the brainstem. So what we do if we see that the blood pressure is off? In short term, how can we regulate this blood pressure? Well, we can do various different things. Of course, we can give the patient medication. That's probably going to fix it in short term. But then they're going to be taking medication forever. We can treat the cause, which would be kind of a smart way of dealing with this change in the blood pressure. Or we can do vasodilation or vasoconstriction, depending on whether the blood pressure is too high or too low in comparison to this set point. So that tells you that this short-term regulation of the blood pressure can really be explained by this homeostatic mechanism loop. Well, the long-term regulation of the blood pressure happens through the kidneys. And that's a little bit more complex process. We have there a direct mechanism, and we also have indirect mechanism, which is a little bit more complex. That's referred as the renin-angiotensin-aldosterone mechanism. You don't need to know it in detail, just as long as you know that there's a direct way and indirect way. So, really, if we want to summarize what did we get out of talking about blood pressure, we want to have a high enough blood pressure so that we are sure that the blood gets to all of the parts where we want the blood to get to. But we don't want to have too high blood pressure so that the small blood vessels, especially capillaries, don't burst. So it's all about finding the balance between these two. And that brings us to the very last thing that I want to discuss with you guys, which is really having a look of various factors that can affect your blood pressure. I mentioned earlier on that your physical state can affect. Some athletes have lower blood pressure. Also, your posture will affect. That's why we try to get you sit down with a good posture when we take your blood pressure. If you're really upset, really worried, really scared, your blood pressure will spike up. Also, if you're sick, for example, fever, that will have an effect on your blood pressure. Your age, sex, weight, health in general, even race, mood, they all affect your blood pressure. So now let's look at two conditions where the blood pressure is not what we are normally expecting it to be. And we're going to start with hypertension. That's when we have high blood pressure. And this high blood pressure is constant. It's sustained. So usually the cutoff values, again, they can vary a little bit, but I think your textbook talks somewhere around 140 over 90 millimeters of mercury for that. Is it bad to have high blood pressure? Well, why would this be dangerous? There's certain risks that come with that. For example, the heart failure, vascular diseases, renal failure, and stroke as well. The renal failure really refers to the fact that when we're filtering blood, the blood pressure is too high and it starts breaking structures there. What we end up finding in individuals who have a very high blood pressure is that their heart muscle ends up having to work harder. So the myocardium becomes thicker and larger. But even though it's thicker and larger, this is not great news for us because it is actually fairly weak and it's fairly flabby. And also the plaque starts building. So even you have a big, thick heart muscle, it's just compensation for that, trying to push that blood out of the heart hard because there's already a high blood pressure, high pressure against of it, which it has to constrict. So that's not great. Some of the risk factors for that, for example, you can inherit likelihood to develop hypertension. Your diet plays a role, especially high fat, high cholesterol food, obesity, age, having diabetes, stress, smoking, all sorts of things that we can, most of those, control through our lifestyle choices. So if you have hypertension and you don't want to jump directly to the medication, consider doing these positive lifestyle changes. Often we do end up finding, though, that there's no clear cause. What causes your hypertension? So really it comes down to these lifestyle changes of managing it. Let's look at another scenario. In this scenario, we have hypotension. So we have a low blood pressure. Low blood pressure is usually classified something below 90 over 60 millimeters of mercury. Usually this is not an issue unless it's so low that the tissues don't get blood. So that blood doesn't travel properly to the tissues where it should be going. Some have argued that this could be even beneficial and be associated with long life and lack of cardiovascular illnesses. The jury is still out on there. There's a little bit of debate out of that. There's various different subgroups of hypotension. So temporary low blood pressure can be, for example, associated when you stand up and you feel a little dizzy when you get up. Chronic hypertension has to do with usually poor nutrition. And this really is associated largely with risk of developing other conditions because you have that poor nutrition. And the acute hypotension is usually an indication of a shock that an individual might be going to the shock. And I do want to talk with you about signs and symptoms of the shock. You know that we in the UK, we call our cops police constables. So we love to make fun of the police constables as well. So we have came up with a mnemonic. Police constables are fat. PC fats. That's the signs and symptoms of the shock. Individual who's going to the shock, their skin is pale in color. Their skin also will feel cold, cool, clammy. Their breathing will be very fast, but it will be shallow. It's not deep breaths. And also their pulse, if you measure that will be fast, but weak. They might appear a little bit agitated, irritated with you. They often complain that they're thirsty, and that means that they're about to go to the shock. I do want you to know the signs and symptoms of the shock. I think that would make a great exam question. So how do you treat someone who's going to the shock? You want to lay them down. The big risk there is that they fall down and they hit their head and we have an injury from that. Also, when they're laying down, we're able to then pass the blood to all parts of the body easier. We might want to raise their feet so that we're directing more blood from the feet to the head, to the brain, which is the part that we really want to support. We really want to make sure that it gets the blood. If there's any constricting clothing, you might want to loosen those. You want to calm the patient. You want to monitor them. And of course, the smartest way to treat the shock would be to treat the cause. But if there is a large blood loss in an individual, it is really likely that that patient will go into the shock. So that's what I wanted to say there. And what we're going to do next, I think, is just kind of talk a little bit about how do we control this blood flow to right places. So we want to have right amount of the blood flow, not too much, not too little, so that it has the time to do the exchange, but that blood also moves off from those sites. So this can be by sympathetic nervous system and hormones. This would be our extrinsic control, but it can also be by the capillary control, so meeting the local needs in intrinsic control. And what I want to do next, we're going to talk about the blood flow in different parts of the body. And this is the very last thing that we'll do. And what you'll find, I'm not going to list, even though you would think that, okay, we're studying blood vessels, and I have not listed you any of the major blood vessels. I'm not going to list for them. I have a really good handout for you that I put together, and that you can use in your exam, in your tests, on my course. So it's a good way of making life easier for you. There's no other way of really learning the blood vessels than studying them. So there's no great mnemonic or just thinking about it and it would make sense. It's just study. So we're going to go through some special adaptations in different parts of the body, and then we're going to have a look at that handout. So what I want to show here, I want us to focus on the skeletal muscles and notice that during normal situation, about 20% of your blood is directed to the skeletal muscles. But while you are exercising and doing physically strenuous activity, this can climb up to 70% of your blood flow being directed to the skeletal muscles. So we're amazingly good at responding the need for the blood if our muscles are working hard. The other thing that I wanted to talk really quickly was our brain. So brain requires a constant blood flow. So if you don't have a constant blood flow to the brain, we notice that there's damage starting really quickly. So that's why CPR becomes so important if someone's their heart is not beating and they're not breathing themselves. So brain cannot handle too high or too low blood pressure. So we're constantly regulating that and making sure that it's the optimal. I want to say a couple of words about the skin as a special adaptation. Skin plays a special role in thermoregulation, so regulating our body temperature. So if we are too warm, we direct that warm blood to the skin where some of the heat will then get lost. You might also start sweating and that evaporation grabs some of the heat with it, and so on and so on. Other thing, that the skin is a great reservoir, great place to store a lot of blood when we're not needing it in other parts of the body. I want to also mention the lungs. We kind of have touched upon that, and we will talk more later on this course. So we want to have low blood pressure within the lungs, because if it's too high, those tiny capillaries will burst and we will start leaking blood to the lungs. That would not be great. So we remember that the pulmonary loop had a low blood pressure for this reason. And finally, we've already talked about the specializations of the heart and how blood is being distributed to the heart through the coronary arteries. So I'm not going to repeat that. So really, if we think of the flow of the blood, that changes in different parts of the systemic circulation depending on the needs. And this is really a nice diagram for us to have a look at. So this diagram shows the speed of the blood in various different parts of the systemic circulation. So here we have the aorta. Blood flows really fast within the aorta. By the time we get to the arteries, it's still really fast. But once we get to the capillaries, that's where the blood flow is the lowest. That's because that was the site of exchange of materials. So we want the blood to move as slow as possible so we can exchange all of those materials within the capillaries. Then once we return back to the veins, the blood starts flowing a little faster, but never quite as fast as in arteries. And then the vena cava that returns the blood to the heart. I think that would make a really good exam question, that let's list the speed of the blood in different parts of the systemic circulation from highest to the lowest. So it would be aorta, arteries, veins, venules, arterioles, capillaries. So it would be a little bit of a mixture. I think that makes a fun exam question. Well, if we map next to this map, the total area, cross-sectional area of these different kinds of vessels in the body, we'll notice that the capillaries are the area where we have largest area for the blood to be at, and vessels have largest area that they cover. Just a couple of final notes. Of course, we remember that the reason why capillaries have the largest area and slowest speed was that we can exchange materials from the blood vessel to the outside of the blood vessel. So how does that happen? That's due to diffusion. So we move on from the high concentration to the low concentration. So it spontaneously flows to that without extra energy, extra effort. Other thing that we notice, of course, we have these fenestrations within the capillaries to facilitate this crossing of the materials. And of course, there might be some amount of active transport. So this is where the various different pumps and vessels for the larger molecules play a role in moving materials in and out of the capillaries. The final thing that I'm going to mention more of as a note for what we'll talk about in the next week's material is going to be two forces that play a role in the blood vessels. The force that's pushing the blood out of the blood vessel or pushing towards the blood vessel wall is going to be our hydrostatic force. So hydrostatic force is all about pumping or pushing the blood out from the blood vessel. Instead, osmotic pressure is the pressure that pushes the blood from outside back in to the blood vessel. And usually these are in balance, so we don't lose, we don't gain blood. If we are ending up pumping a lot of blood or components of the blood, especially the plasma, out from the blood vessels, we end up with edema. So edema is this swelling because there's too much now interstitial fluid. So material leaking out of the blood vessels to that side. How can you tell if someone's just fat or if they have an edema? You push it and you have a look. If it's in an edema, it doesn't bounce back immediately. It stays kind of depressed for a while. And final note, this is very, very last slide, I promise. Important things to note about the vascular system. I think that this summarizes nicely. Remember that arteries run deep within the body. These were the vessels that are right leaving the heart. They have high blood pressure. This blood is pumped with high force. So if there was a cut, they would bleed like crazy and you would die really quickly if we cut this. So that's why the arteries are kind of stored deep in your body, protected by other tissues and other structures. Whereas the veins are going to be fairly superficial. So there's low pressure against those vessels. So they are not squashed closed. They are only squashed so that we can move the blood, but they're not going to be fully closed. The other thing to notice is that veins are going to have more connections between each other. So there's large networks of veins. They find an alternative way for the blood to go if one path doesn't work. So there's a lot of anastomosis there. And finally, I want to mention that the brain, just as well as intestines, they need to have some unique drainage arrangements, especially in terms of the intestines. Most of the blood travels to the liver. And that's so that we can store the energy from that blood that's being taken, the energy that the blood has picked up from the intestines, from the digestive system. It goes to the liver first so that we store that energy. And that really is all that I wanted to say in terms of this chapter, but I do want to show you, before we completely stop revising the material, I want to show you the diagrams that I mentioned earlier on. So when you go on Canvas to your this week's course, you will notice there that I've uploaded a set of PDF files, and I think they're called something like I want to say vessel diagrams or something sounding like that. And these actually provide many different kinds of diagrams that show the major arteries and major veins that you might need to know. So this is very technical or diagrammatic. We would have our heart located here. And out of the heart, we have our aorta coming. You remember we had our coronary arteries initially, and then we have these branches of the aortic arch. And then it travels down to the thoracic aorta and abdominal aorta and divides wherever it's needed. Well, if you don't love this kind of a diagram, it seems too theoretical for you. I've done the same diagram on this slide or this sheet for you by superimposing these important arteries on a human body. So that might be useful if you don't like the earlier on. You still see the heart and how all of them start from the heart. Well, if we continue, I have specific diagrams, for example, to the head and neck. So here we see heart would be located here, aortic arch, especially the supply to the head and neck and to the brain from both left and right side. And then the blood would go also, of course, further down to the rest of the body. If you don't like that diagrammatic representation, I've got it for you kind of superimposed again on a diagram of a human body. This is where the heart is. And then from the aorta, these branches that go to the head and neck. And the important arteries that you need to know. Same for the brain. So there's some important ones that you might want to know if you're interested on the brain and brain anatomy and physiology. I've done similar for the upper limb. So you would have your heart again here, your aorta with aortic arch, and then we look at the head, neck, and upper limb all the way to the fingers. The most important arteries that you might need to know. If you didn't like this diagrammatic, schematic representation, I have it superimposed on a traditional diagram. Again, heart and then various parts of the upper limb, all the way to the fingers, showing the important blood vessels and names of those. Same goes for the abdomen. Again, very diagrammatic. Heart would be here, and the blood goes from the aorta down to the thoracic aorta and then abdominal aorta. And all these branches that come out of there, all the way to where it heads to the legs. Well, if you don't like that, I have more traditional drawing in multiple levels, so you can see behind the organs where we start seeing these branches. And of course, I have done the same for the lower limb all the way to the toes here. And that would be, of course, the mirror image on the other side. Again, if you prefer the same superimposed on a traditional diagram of a leg. So I often focus on the arteries because we find arteries from one individual to another being very, very, very similar. They're located at the same place and they're very predictable. Major veins are also predictable, but at large, veins are less regular. So you end up finding that veins can be a little off from where you expect to find them. So they can be a little bit more challenging to identify. But again, I've mapped those for you as diagrams, but also on top of the human body. For all of the parts that we had a look of earlier on, head and neck, brain, upper limb, abdominal area, thoracic area, and lower limb. So there's a bunch of these diagrams. I encourage you to have a look of those. If that's something that you think that you want to save or you want to download for yourself for use on this course, but also on the future. So what we've done, we've covered what I wanted to say about the key points of the chapter on the blood vessels. And really the last thing that I have left to do is to remind you that you have tutoring available through the Cranium Cafe, the virtual tutoring services here. I'm also available, like I already mentioned. I've had a bunch of meetings with various students on the past couple of weeks. I'm super happy to set those up. My job is to help you. So if there's anything in terms of technical issues that you need help, or if you have questions about the actual content material, I am here to help you. Just let me know. I'll usually be able to organize you an appointment, if not the same day, then the next day. I will work to make that happen. This would be the time when I would ask that if you have any questions that I can address. If you don't have questions, really we have covered everything that I set out to do on this week's material. Notice that these special circulations I leave for you to revise based on those handouts of those special diagrams of where the major arteries and veins go. If you don't have questions, that's all I have to share with you today. If you have questions, I'm going to be here. I'm happy to answer. I'm also happy to stop the recording if you don't want to ask them on a recording. But that's all from me for today. I really appreciate. Okay, blood anatomy video is not displaying. Let me look into that. I'll fix it right after this session. Blood anatomy video. I'll have a look. I think I know what's the issue there. And I'll have a look of that. I think it's just that I need to click a button so that it opens in a different window. Is there, okay, thank you, Savannah. Do you have any other questions that I can help you with? I'll have a look of that blood anatomy video and I'll add it to my announcement that it's working. But if you give me, say, 20 minutes after this session, it should be working after that. If there's anything else, just ask away. If not, I hope you have a really good rest of the night. Absolutely, your lab report. So do you mean your lab practical score? Exam, yes. So let me just pause the recording. So we're back recording. So what I was speaking with one of the students was that on this week's practice anatomy video on the blood vessels, that video link for some reason has gotten broken. And that's a video link that actually my colleague has an ownership over. So she has prepared all those practice anatomy videos. So I'm going to have to ask from Professor Greer that what's up with that video, and I will update you on the announcements. So if you now go and try to open up the anatomy video for the blood vessels, where she goes through those important structures like she goes in every chapter, it's likely that that video will not work. You're not doing anything wrong. For some reason, it seems that YouTube has either taken it down or she might have turned it private or there's happened something. So I will find out from her. And I think that by tomorrow, I should have an answer. So I send it with the announcement for you so we can have a look what's going on with that then. So without further ado, that's all I have for this video. I really thank you for joining me once again. I think we've covered a lot of ground. I know it was a little longer of a session. I hope that everything made sense. And I really, like I said, I encourage you to have a look of those special circulations from the handouts that I have provided. It doesn't mean that we didn't cover them in the lecture videos, that it wouldn't be important. It's just something that you really just have to memorize. So on that note, that completes our week 6, looking at the blood vessels and finishing the cardiovascular system. I thank you for joining me. I hope you have a very good rest of the week. Remember to contact me if there's anything at all that I can help you with, any questions, any trouble, whether it's technical or related to the content of this chapter or any other chapter, whether it's about exams, practicals. I am here to help you. I want you to succeed and reach the very best score that you can get. So thank you. Have a good night, and I'll see you next week. Keep an eye of the announcements that are coming up. Probably tomorrow. Thanks. Bye.